So hi, I'm Kasia. I'm from Cardiff University and I'm just got into my second year of my PhD. So I'm looking at a cystic fibrosis bacteria called Burkholderia multivorans, and I'll be focusing more on the genomics kind of side of my project for this presentation. So it's going to let me go to the next slide, which is not. There we go. So first of all, what is cystic fibrosis? So it's a recessive genetic condition which is caused by a mutation in the CFTR gene. It's mainly found in the Caucasian population and is passed from parent to child. It affects around 1 in 2,500 births, with 10,000 people in the UK affected. So why is this important? Well, basically, it can cause this thick, sticky mucus in the lungs. And long-term issues mean that you can have bacteria and viruses colonizing the lungs and causing these infections. What we want to do is try and control these infections and look at how they colonize to try and help basically prolong life. So what's quite interesting is you get this polymicrobial infection. This is where you have more than one organism present in the lung. So I'm not going to go too much into this because this is one half of my project. But basically, I work on this tiny little bit down here, which is known as the Burkholderia cepacia complex organisms. My work focuses mainly on Burkholderia multivorans, which is part of this. So what are the BCC? Well, the BCC are part of this genus called Burkholderia. It's made up of both plant beneficial and pathogens, so things that cause infection. What I want to look at is this one called Burkholderia multivorans, and this is part of the one that causes the infection. So, the aims and objectives of my PhD. Well, I have two really. I have this one I mentioned, which is to model the interactions of different pathogens, but what I'll be mostly focusing on is to look at the pathogenesis and phylogenomics of Burkholderia multivorans, looking at its population biology and trying to develop novel therapeutics against this organism. So I have two sort of arms of my research. So one is this phenotypic characterization, so looking at things mainly in the lab, looking at the pathogen alone and then when mixed with other pathogens. So these are different assays looking at its motility, its biofilm formation, protease reduction and more. What I'll be talking about today is the genotypic characterization. This is using whole genome sequencing with phylogenetic trees and genome mining. So I know this looks a little scary, but basically this is what is the major part of my research at the moment. So what I originally did, and I'm not going to bore you with loads of different trees because there's so many, I took this panel of 49 Burkholderia multivorans from the collection at Cardiff University, and I put these through whole genome sequencing. I essentially found this initial split of a tree where it looked like it went into two evolutionary lineages. What I went from there is I downloaded over 200 different genomes from publicly available databases and made a massive tree to show this robustness. What I'm showing here is my final 77 isolates, strains. This is where I've taken relevant reference strains and my initial 49, and I've put them into some phylogenetic analysis and genomic analysis for you. So on the left, what you'll see is something what we called average nucleotide identity analysis, that's ANI. This essentially looks at the relatedness between the different species, the different strains, sorry. So what we call down here is what I've denoted as lineage 1. So the darker the red colour, the more related they are. So in this lineage 1, they're all quite related, very, very well related, over 99% identity. I've then looked and I can see the second grouping here. And what we've said is we think there's also two subgroupings within this. Obviously, this is quite, I would say it suggests it, but it's quite subjective still. So what we wanted to do was to go and look at this in whole genome sequencing. this is what is on the right. So what I did was I used a maximum likelihood tree and used the core genes of my 77 isolates to build this tree. This was using a RaxML software in command line. So basically what you can see is where I've highlighted lineage 1 here, which groups off, and that's shown down here, and this lineage 2. You can see that there's the definite distinct lineages between the two. What I've done is I've then color-coded these based on the three different groupings that I've talked about. So we have got this other one, which is an outlier, which I've denoted just as other. So this is one of the isolates from the collection. It is a cystic fibrosis isolate, and we don't know why it sits on its own, and it doesn't seem to have anything that suggests this so far, but that's more things that we'll look at down the line. So just going off this, these aren't all cystic fibrosis isolates. So what I've done is I've color-coded them accordingly. The ones that are most important are the ones in black, which are cystic fibrosis, and ones in green. So what you can see is down here, these ones in green that are sat in what I'm calling lineage 2A are all environmental isolates. There is one that sits up here in lineage 1, but further genome mining would underpin what happens with this. And all those asterisks are those that have been sequenced as part of this study. So you can see that it's quite a nice spread. What I went to do more of is to look at the statistical analysis to confirm the presence of these two lineages. What you can see from the statistical analysis is it provides a certainty that there is this split in the two lineages, and also one for the three. So this is lineage 1, lineage 2, 1, 2A and 2B. And you can see that lineage 1 has quite a close average nucleotide identity compared to lineage 2. However, the significance shows that there are definitely 3 groupings. What we wanted to find out though is what differences there are between these groupings, and this may give us more of an idea of why maybe Burkholderia multivorans resides in the lung compared to another one. So what I did was, from one of my genomics outputs, I looked at the different number of genes in various categories. So what I've done is some genome mining to look at different virulence genes. So virulence genes are things that which help the pathogen cause disease. So what I've looked at is the overall gene numbers of all 77 isolates, and I've just tallied them up in these different categories. This is just a brief overview to what I'll be talking about next. So what I've done is I've taken some of these categories and then I've looked at them between the lineages. What you can see is there's some significance, especially in biofilm and adhesion-related genes and biocide resistances between the two lineages. These are ones that we'll look at further, but at the moment we've only just started touching on this in the research, so not too much is known at the moment. I then took this further and took it into 3 lineages to have a look at a bit of differences. There are some differences between the two, but the majority is between lineage 1 and lineage 2 overall, not between the sublineages 2A and 2B. So what we can say is there is a lot of genetic variation. Why there is, we don't actually know. But a lot of these infections are either chronic or they can be transient, which means that they come and they go. So understanding this may help us understand why the different strains are colonizing better than others, and to say, wait, why maybe lineage 1 is better at colonizing than, say, lineage 2. So hopefully this gives you a bit of a small snippet of my research. This is more the genomics half, like I said, and it's the smallest amount of my PhD so far. So thank you for listening, and I hope that you enjoyed that.
Cool, that was fantastic. Thank you very much. So I guess we'll use the opportunity to open the floor to questions now, if people have any. So I guess if I start off then. So you were saying that you're looking at the bacteria you're looking at and you're grouping them into different lineages.
Yeah.
So I guess cystic fibrosis is a very clinical thing. So what's the main advantage, I guess, of grouping bacteria into different groups like that and finding these different lineages?
So basically, for this one, what you want to do is, like I mentioned, some cause more chronic infections. By understanding this population biology, what you're going to understand is why maybe one is going to be more persistent and then therefore can interact with other organisms and essentially colonize the lung and cause death, which we don't want to happen. So understanding this may help us intervene, and understanding potentially any antibiotic resistances or anything like that, if that makes sense.
Yeah, that makes perfect sense. And we actually have another question from the chat from the other presenter, Dr. Yenel herself, if I'm pronouncing that correctly. You can correct me before you talk. So the question is, do all cystic fibrosis patients have the same bacteria?
No, so they don't. And this is what I started to touch on, but I didn't go too much into because it's basically a minefield and I could talk for ages on it. So what I'm looking at is only in a tiny amount of patients. So basically, Burkholderia infection is one of the things that happens sort of last. It's one of those things that you get after quite a bad chronic infection, and it leads you essentially to death. And this is usually seen in later stages of infection and older ages. But it completely depends on the individual. You don't tend to get, say, 2 Burkholderia infections at once. You wouldn't get a Cenocepacia species with a multivorans. But what you could get is sort of a Burkholderia multivorans with a Pseudomonas aeruginosa, for example. And this is where, in part of my research, we're looking at the interactions between these. And that's the other arm of what I'm doing as well. So it completely differs patient to patient, really.
Perfect. Thank you very much. Do we have any more questions? Oh,
Yes.
Oh, of course, it's you, Anna.
Yes, sorry. I just wanted to ask, you've done a lot of predictions with the statistics of the different genes.
Yeah.
Do you have some of these predictions, like, do you have an idea from like the lab work whether this tallies up with what you predict, like the biofilm formation and so on?
Yeah, so the reason I didn't talk about that is because there's absolutely no differences in them.
Okay.
So obviously that is still a result. We can't really say when it's not significant, it's not a result. But basically what we saw at phenotypic level is basically nothing, but when you delve into genome mining, that's where you see all the differences between the 2 lineages, and that's what we're finding more interesting. So
Really
yes
good.
and no.
A lot of work and a very good presentation. Thank you.
Thank you.
So, Kasia, I've actually had one last question submitted to me, which I know is everyone's favourite if they work in Burkholderia. Where do these Burkholderia come from? Are they just in the lung, or are they from the environment? Where do multivorans come?
Don't you? Oh, I hate you.
I can only do. I'm pandering to the public.
Okay, so we don't actually know. That's the thing. They're thought to be from the environment, and there's suggestions that they come from the environment, but we don't really know. We know that they don't spread person to person, but that's about it, really. There's no defined niche or anything like that currently. But the reference sequence was essentially isolated from the environment. So